Hello everyone, let's talk about our skeletal muscle physiology. For our skeletal muscles to contract, three major steps must take place. A bunch of events that need to take place at our neuromuscular junction. Then steps known as the excitation contraction coupling. Then the cross-bridge cycle. What is a neuromuscular junction? So, as I said, our skeletal muscles, they are voluntary. So we have conscious control over them. And so when we want to, let's say, flex our arm, flex our leg, we gotta send an electrical message to our leg muscles, to our arm muscles. Those electrical messages are known as action potentials. So you can see right here, here is our muscle fiber or our muscle cell, and here are the neurons right on top of our muscle fiber. And this is the ending part of our neuron known as our axon terminals. And you can see the red arrows are representing the action potentials, those electrical messages moving their way down to the neuron so that they can reach the muscle fiber. The events that will happen right here at the axon terminal is what's known as the neuromuscular junction. Neuro meaning neuron, muscular it's your muscle, junction it's the connection between the two. So let's go ahead and zoom in on the events that will happen right here. So right now we are going to explain what's going to happen at the neuromuscular junction. So this is a zoomed in image of the axon terminal, and this is representing your muscle fiber or your muscle cell. On top of the muscle fiber, we have the layer known as the sarcolemma, which is kind of like the plasma membrane of a cell. So the sarcolemma is right here. Now, for us to go ahead and start contracting our muscles, we gotta send an electrical message, we gotta send an action potential, and that action potential is going to make its way all the way down to the axon terminal. Now, an action potential running towards the axon terminal, that action potential is depolarizing the inside of your axon terminal. So that the depolarization state is going to open up voltage-gated calcium channels. Those voltage-gated calcium channels, once they open, calcium is going to start entering into the axon terminal. Once calcium floods inside of the axon terminal, the calcium is going to trigger those vesicles right here carrying neurotransmitters. It's going to trigger them to go ahead and release the neurotransmitters via the process known as exocytosis. And the neurotransmitter is going to flood this region right here known as the synaptic cleft. The important neurotransmitter that's going to help your muscles contract is a neurotransmitter known as acetylcholine. So acetylcholine gets ejected out of the vesicles into the synaptic cleft. Once acetylcholine is ejected out, it's going to bind to specific receptors found on the surface of the sarcolemma. So on the surface basically of the muscle cell, specifically on the sarcolemma layer. Those are the events happening at the neuromuscular junction. Once the acetylcholine binds to the receptors on the sarcolemma, we are going to go ahead and start the excitation contraction coupling. So right here you can see action potential running down to the axon terminal, causing the events that we've just talked about. Acetylcholine gets ejected out. Once acetylcholine is ejected out, it will bind to the receptors on the surface of your muscle fibers, specifically on the sarcolemma. Once it binds to the sarcolemma, those are going to be the steps known as the excitation-contraction coupling, which we will talk about it in detail. But the excitation-contraction coupling is going to generate another action potential. So let's go ahead and zoom in to check out how is that going to happen. Let's talk about what this image is showing us and talk about the details that we see, and then we will go ahead and talk about the steps that are going to take place for our muscles to contract. You can see right here the axon terminal. So this is where we have the neuromuscular junction, the connection between the neurons and our muscle fibers or our muscle cells. And you can see right here on the surface of our muscle fiber, we have our sarcolemma, and you can see that the sarcolemma starts to dip inwards, creating those T tubules. On the surface of the T tubules, we have receptor channels known as dihydropyridine, or DHP for short. And you notice that those DHP channels, they are actually connected to another channel right here known as RYR, or ryanodine receptors, and those ryanodine receptors are on the surface of the sarcoplasmic reticulum. The sarcoplasmic reticulum, this is where we store our calcium ions that are needed for our muscles to contract. So you can see that those two channel receptors are currently closed, and you can see that they are connected to one another. And then over here we have our myofilaments. So you have your actin and your myosin filaments, which are going to be those protein filaments that would help you contract your muscles and relax them. So as we said, for our skeletal muscles to contract, we have to go through several steps. The first major step is the events that are going to take place at our neuromuscular junction, which we already explained. And we said that acetylcholine is going to be ejected out into that space right here known as the synaptic cleft. Once acetylcholine is ejected in the synaptic cleft, we can go ahead and start our excitation contraction coupling steps. Acetylcholine, once it's in the synaptic cleft, you notice that on the surface of the sarcolemma there are receptors, acetylcholine receptors. Acetylcholine binds to these receptors. Once acetylcholine binds to those receptors, sodium ions are going to start flooding, and they are going to start basically entering into your muscle fiber or your muscle cell. Now, why is sodium going to be entering into the cell and not exiting? Because as we said, we have way more sodium ions outside of our cells. And so diffusion is going to allow those ions to move from the high concentrated area, which is in this case we have more sodium outside, into the lower concentration area, which is inside of our muscle cells. Now, sodium ions are carrying a positive charge. So when you have all of these sodium ions, which are positively charged, flooding the inside of your muscle cell, an electrical message is going to be triggered. We refer to it as the end plate potential. Now, why end plate potential? Because right here, this region right here, where you have the junction between the neuron and the sarcolemma, we refer to it as the motor end plate. So that electrical message that's going to be triggered is going to be known as the end plate potential. Now think of your end plate potential as our graded potential. For an action potential to get triggered, remember we talked about it in our action potential video, a graded potential must reach threshold to trigger an action potential. So our end plate potential must reach threshold for an action potential to be triggered. Let's say that the end plate potential did reach threshold, then an action potential is going to be triggered, and the action potential is going to run down the T tubules. Once an action potential runs down the T tubules, those receptor channels right here, they are actually going to change their conformation. And notice how those DHP receptors are attached to the RYR receptors. So once DHP changes its shape, it's going to pull open the ryanodine receptor. If those receptors are pulled open, what's going to happen? Calcium is going to start flooding the inside of your muscle cells. So we can see right here, those ryanodine receptors, notice how they opened up. Once they open up, calcium rushes outside. When calcium is rushed outside, calcium is going to bind to a specific protein on your actin filaments known as troponin. And once calcium binds to troponin, then we are going to go ahead and start our cross-bridge cycle. We are going to talk about the details of our cross-bridge cycle in just a minute, but as long as we have calcium bound to our troponin protein on our actin, your muscles are going to start contracting, and they are going to keep contracting. Now, for us to stop our muscle contraction, well, we have to return the calcium back into the sarcoplasmic reticulum. And so there is a pump right here known as the calcium ATPase, and the calcium ATPase pump is going to pump the calcium back into the sarcoplasmic reticulum. Once the calcium goes back into the sarcoplasmic reticulum, then your actin and your myosin filaments are going to let go of one another, and we can start our relaxation phase. So right here, this is showing you the sarcoplasmic reticulum where we have all of the calcium ions stored. So we said that first we're going to get that end plate potential. If it reaches threshold and an action potential gets triggered, that action potential is going to run down the T tubules, changing the conformation of those DHP receptors and opening the ryanodine receptors, causing calcium to flood out. Once calcium floods out, it's going to bind to the troponin protein on our actin filaments, and cross-bridge cycle can go ahead and begin. So right here, let's zoom in to explain what's going to happen with our cross-bridge cycle. We already talked about the anatomy of our actin filaments in the previous skeletal muscle video. So we said that our actin filaments, we've got those subunits, and those subunits have active sites. And we said that there is a protein, tropomyosin, that's blocking those active sites. And as long as tropomyosin is blocking those active sites, then you're not going to have any contact between actin and myosin, and your muscle is relaxed. For us to begin our contraction, after we go through the steps of the neuromuscular junction and the excitation contraction coupling, and we said that at the end of excitation contraction coupling, calcium gets released. Calcium binds to troponin. When calcium binds to troponin, tropomyosin is going to unravel, exposing those active sites. Once those active sites are exposed, the myosin heads are quickly going to grab on to the actin filaments. So right here, you see that they've already grabbed on, and after they grab on, they go through what's known as the power stroke. Notice how the myosin head bends? So it quickly bends, and when it pivots or bends like that, it's going to pull the actin filament towards the M line. We said that we go through something known as the sliding filament theory, where we already talked about that during the sliding filament theory, the actins are being pulled towards the M line. Now we see why they're being pulled. The reason why they're being pulled is the myosin heads are going to grab on to those actin filaments, and you can see that they're going to start pivoting and they're going to start pulling those actin filaments towards the M line, causing your muscles to contract. Now, for your muscles to relax, those myosin head needs to let go of the actin. The only way your myosin head is going to let go of your actin is if ATP energy binds to the myosin head. Once ATP binds to the myosin head is going to let go. Now, after ATP binds to the myosin head, notice how the head is still bent. Now we need to allow the head to go back to its normal position. And so to do that, ATP breaks down, or we refer to it as ATP hydrolysis. Now there is an enzyme that's actually going to help us with the hydrolysis of ATP, and that enzyme is known as myosin ATPase. So myosin ATPase, what it does is it helps break down your ATP into ADP plus a phosphate, an inorganic phosphate. ATP stands for adenosine triphosphate. So ATP has 3 phosphates bound together. Myosin ATPase is an enzyme that will help break down ATP into adenosine diphosphate, so 2 phosphates, and then a phosphate on its own. We refer to it as the inorganic phosphate. Now, myosin ATPase helps break down ATP. And why is that important? Because when ATP breaks down, notice how right here, the head returns back to its normal position. And why is that important? Why do I want my myosin head to return back to its normal position? Because once it goes back to its normal position, it can grab on one more time to your actin filament, and we can repeat the CrossBridge cycle one more time. If your myosin heads do not go back to the normal position, then we're going to have a problem. And so if you think about it, the more myosin ATPase we have, the faster our muscles are going to contract, because the faster those myosin heads return back to their original position, then they're going to be able to grab on, and we're going to have another contraction. So as you can see right here, once it goes back to its normal cocked position, it grabs on and the cycle begins. And you can see that right here at the power stroke, the ADP and the phosphate, they are also going to be released. You can see that the power stroke goes ahead, starts pulling the actin filaments towards the middle. One more time for your myosin heads to let go. A new ATP is going to come and bind, and the cycle keeps going. That's known as the cross-bridge cycle. Myosin heads grab on to the actin filaments. The myosin heads pivots, pulling the actin filaments towards the M line. For the myosin heads to let go, ATP must come and bind. To bring the myosin head back to its cocked position, ATP hydrolysis will take place by the help of the myosin ATPase enzyme. Once it's cocked back and you still have calcium around, which means those active sites are still available, the cross-bridge cycle will take place one more time. For us to stop that cross-bridge cycle, we must push back the calcium back into our sarcoplasmic reticulum. Once calcium is pushed back into the sarcoplasmic reticulum, tropomyosin is going to block the active sites on your actin subunits. And the subunits are basically right here. So if those subunits, if those active sites on our subunits are blocked, then myosin is not going to be able to bind, and your muscle will go ahead and relax. When a person is dead, after a person dies, no more ATP is going to be released. And so the cadaver is going to, the dead body is going to experience something known as rigor mortis, where a few hours after death, the muscles of the cadaver is actually going to stiffen up. The muscles are actually contracting. How come? After death, calcium is going to start getting released out of the sarcoplasmic reticulum, out of storage units in your cells, in your blood, and those calcium ions are going to bind to troponin. Tropomyosin unravels, active sites are exposed, and basically your muscles will contract. But the thing is, since you do not have any ATP, the myosin heads are not going to be able to let go. And therefore, rigor mortis causes the muscles to stiffen up for a few hours. Then eventually, as everything starts to deteriorate, the muscles are going to relax one more time. They can actually use rigor mortis to figure out how long has this person been dead for. So they can tell exactly kind of range when did that person die. And rigor mortis is affected by the temperature you find the cadaver at. So cooler temperatures, rigor mortis is going to be much slower compared to if the body was found in a region that's pretty warm. Rigor mortis takes place pretty fast. This image right here kind of shows you that for your muscles to contract, first we have to trigger an action potential, and the action potential runs down along like your muscle fiber, and it goes deep into the T tubules, and then we have to go through the excitation, contraction, coupling, and then your muscles will go ahead and start contracting by going through the cross-bridge cycle. So this image right here shows you that during this period right here, known as the latent period, your muscles are not going to be contracting during that latent period. The latent period is basically the steps happening during our excitation contraction coupling. So the action potential is going down the T tubules, the sarcoplasmic reticulum is releasing calcium by the channels, the ryanodine channels opening up. So during the latent period, all of these steps that we've already talked about, they're taking place so that your muscles can go ahead and begin the cross-bridge cycle. Once we hit this region right here, known as the contraction phase, then we know that cross-bridge cycle started taking place. So the contraction phase is basically the beginning of the cross-bridge cycle. And then for the relaxation phase, we said that for us to relax, myosin heads have to let go, and that happens when ATP binds. But then to prevent the myosin heads from reattaching, we have to push those calcium ions back into the sarcoplasmic reticulum. Then once calcium goes back, tropomyosin goes ahead and returns back to its normal position or normal look, blocking the active sites on the actin, and your muscles relax.